How?
Polyamine metabolism impacts T-cell dysfunction in the oral mucosa of people living with HIV. RNA sequencing of oral gingival mucosa and salivary metabolome analysis show dysregulation of arginine and polyamine pathways in PLWH. Hey everyone, welcome back to our channel. Let's talk about RNA sequencing. This is a powerful technique that allows scientists to analyze the entire transcriptome of a cell, which means we can see which genes are being expressed and how they might be affected by different conditions, like HIV infection. Moving on to the salivary metabolome analysis, this is where things get really interesting. Metabolomics is the study of small molecules, or metabolites, in biological systems. By analyzing these metabolites in saliva, we can get a snapshot of the metabolic changes happening in the oral cavity of individuals living with HIV. Using global metabolomic profiling of saliva combined with transcriptional profiling of the oral gingival mucosa, we first identified metabolic alterations in CR-treated PLWH versus uninfected controls. We combined the transcriptomic and metabolomics datasets using a combined computational approach that annotates gene expression changes associated with alterations in metabolite pool sizes. Linked gene/metabolite associations were subsequently mapped to pathways using MetaboAnalyst and Metascape. Metabolite identifications were made with Compound Discoverer 3.1 SP1 and EMSMean2 software. MS1 and MS2 spectra were matched using a mass tolerance of M/Z equals 0.1. The raw data were acquired and aligned using the Compound Discover based on the M/Z value and the retention time of ion signals. Ions from both ESI- or ESI+ were merged and imported into the SIMCA-P program, version 14.1, for multivariate analysis. Further data processing was performed by using DecoID version 0.3.0, to deconvolute chimeric MS2 spectra and increase the identification rate. Metabolite identifications were made with level 2 confidence according to the Metabolomics Standards Initiative 9. Metabolite changes were considered significant if they reached a threshold of log 2 FC equals 1, p less than 0.05. Using these metabolites and gene lists, we examined pathways significantly altered by HIV infection, group B, N equals 40, compared to uninfected controls, group A, N equals 26. The enrichment analysis indicated a widespread metabolic reprogramming within the oral cavity associated with HIV infection. Partial least squares discriminate analysis, PLSDA, and variable importance in projection, VIP, score plots are shown in supplementary. Figure 1. Alterations in metabolite classes included amino acids, energy metabolites, lipids, and carbohydrates. Together, these changes were reflected in the top enriched pathway hits that include amino acid utilization, arginine and proline, tryptophan, and branched-chain amino acids, arachidonic acid metabolism, and the use of carbohydrates, Figure 1a, b. Activated immune cells have previously been shown to upregulate glucose utilization to support proliferation, differentiation, and effector functions such as cytokine secretion 10. Therefore, we examined the changes in the expression of glycolytic genes in PLWH versus controls. Enzymes that control glycolytic flux including phosphofructokinase, PFKFB3, are strongly upregulated by HIV infection, log 2FC 2.68, P is equal to 1.15 times 104, supplementary. Figure 2. Several transcriptional regulators of hypoxic and glycolytic responses and responses to glucose deprivation such as hypoxia-inducible factor 1, HIF1, alpha and DNA damage-inducible transcript 4 proteins, DDID4, were also enriched in PLWH and gingival mucosa. Interestingly, glucose was also increased in PLWH while the immediate downstream intermediates glucose 6-phosphate and fructose 6-phosphate was decreased compared to healthy controls, supplementary figure 3. However, no significant changes in glycolytic and TCA metabolites, or lactate, were noted in PLWH, supplementary figure 3. Arachidonic acid is also a key lipid mediator driving inflammatory responses 11-12. Our transcriptomic and metabolomic datasets were significantly enriched for hits in this pathway, supplementary figure 2. Therefore, we examined enzymes and metabolites involved in the production of eicosanoids and leukotrienes within the oral mucosa. Arachidonic acid is significantly increased, P equals 0.049, in saliva along with the eicosanoids, 5-hydroperoxyeicocetetraenoic acid, 5-pete, and 12-hydroxyecocetranoic acid, 12-heat, 13. Increased levels of 5-pete and 12-heat were also consistent with the upregulation of their biosynthetic enzymes arachidonate 5-lipoxygenase, ALOX5, and CYP4F3 were detected in the transcriptomic analysis. Moreover, we detected perturbations in the tryptophan/kynurenine axis in PLWH. This pathway consumes tryptophan to produce molecules such as kynurenine that regulate immune cell function 14. Kynurenine also functions as a ligand for aryl hydrocarbon receptors, the activation of which promotes TREG differentiation 15. Although heightened tryptophan catabolism and kynurenine levels would be consistent with the TREG enrichment that we showed previously in PLWH4, we found an opposite trend, including upregulation of kynurenase, moderately lower levels of kynurenine, and increased levels of tryptophan in saliva in oral mucosa of PLWH, supplementary figure 2. Based on the pathway enrichment analysis data, we further examined other pathways that may be related to immune activation in the oral mucosa. We subsequently examined gene/metabolite relationships driving the alterations in amino acid metabolism by mapping the dysregulated genes to associated metabolites, log 2FC cutoff equals 1. Using metabolomics computational tools, including MetaboAnalyst and DecoID16. From this analysis, we identified key nodes altered by HIV infection that are involved in the production and use of amino acids including arginine, figure 1c. To determine age-dependent changes. In control, Group A, and PLWH, Group B, we stratified the groups based on their age, A1 and B1 aged less than 60 and A2 and B2 age greater than 60. HIV-infected individuals showed significant downregulation of several enzymes involved in glutamate utilization or production, including 5-oxoprolinase, APLA, log 2FC equals -1.64, P equals 0.016, and alanine aminotransferase. GPT2, log 2FC equals -1.64, P equals 0.007, independent of age stratification, figure 1c. Reduced expression of GPT2 impacts the production of alanine from pyruvate and is consistent with alterations in the activity of this enzyme. Alanine levels were significantly increased, while pyruvate is decreased in the saliva of PLWH. Interestingly, individuals with HIV showed increases in arginine in saliva while the pool size of citrulline was decreased compared to healthy controls, figure 1c. This led us to examine nitric oxide and ornithine pathways. Several enzymes including two enzymes that play a role in polyamine synthesis, spermidine/spermine N1, acetyltransferase, SAT1, and ornithine decarboxylase, ODC1, show increased transcription in PLWH. The precursor of polyamines, ornithine is also decreased in PLWH, while ODC1, the polyamine putrescine and EIF5 a transcript downstream to putrescine were elevated relative to healthy controls indicating that there may be increased polyamine synthesis in the microenvironment of the oral mucosa, figure 1c. Because ODC1 was recently shown to instruct Th subset Fates 517, we hypothesized that HIV-induced altered metabolic states could impact TREG/Th dysregulation in oral mucosa of PLWH4. Consistent with the upregulation of ODC1 and EIF5A transcripts in PLWH, figure 1c, flow cytometry analyses also revealed heightened expression of these proteins in CD4 plus T cells of PLWH individuals when compared to uninfected controls, fig. 1d gating strategy, supplementary figure 4. Altogether, the oral mucosa of HIV-infected patients provided an important insight into how polyamine metabolism may be relevant to altering T cell functions in mucosae. The researchers found that there's a widespread metabolic reprogramming associated with HIV infection. They used some really cool computational tools to analyze the data and identified significant alterations in pathways related to amino acids, energy metabolites, lipids, and carbohydrates. One of the key pathways that stood out was the arginine and polyamine pathways. They found that individuals with HIV showed increases in arginine in saliva, while the pool size of citrulline was decreased compared to healthy controls. This led the researchers to examine nitric oxide and ornithine pathways, which play a role in polyamine synthesis. What's really fascinating is that they also looked at T cell dysfunction in the oral mucosa of people living with HIV and found a link to polyamine metabolism. This could have important implications for understanding how HIV infection impacts T cell functions in mucosae. Thanks for tuning in, and don't forget to hit the like button and subscribe for more science content. See you next time.
PharmaPal.